Good morning, everyone. I welcome you all to this 20th day of CSIR Life Science question and answer discussion session. Today, we are going to discuss about the methods in biology. Okay, as per our schedule, past 2 days and today is for part C question discussion on methods in biology. Today is very short section because I'm having some another meeting on 9 o'clock, so it's just one nearly 8 questions are there. We will going to discuss in the session. Okay, first question: choose the correct question of event in next generation sequencing. So we want to understand clearly about the next generation sequencing. If you are a biotechnology student, you may know about the next generation sequence because in the genetical mapping, in the genetical analysis, we studied in the biotechnology. Some of the medical and biochemistry people are know about this concept. So in this next generation sequencing, one thing we want to understand: why is it normal integrated process? Okay, this next generation sequencing is integrated process. Okay, Sanger sequencing method and sequencing techniques are there. Various maximum Gilbert method, Sanger sequencing method, and some of the pyro sequencing. All the sequencing techniques are there. Apart from this, why next generation sequencing is very important? It's why different. That is a thing is we want to know. This next generation sequencing, we can compare the larger quantity of sequences. It can helpful for the it's a large quantity of nucleotide. Lengthier nucleotide can be sequenceable. First is the step. Each and every step is important in the this DNA sequencing technique. In this, correct order is answer C. First, we want to extract the DNA. That is the first step. We want to extract the DNA. Then stringing. Okay, stringing means it's one kind of the process which make a flexible DNA for the sequencing. Because during the sequencing technique, that should be in a flexible manner. If it is in a very sticky mode, we can't uh, able to get a proper sequence. So they are cut these small pieces. Okay, that is the step here. So people, for the our DNA sequencing efficiency improvement, we want to cut the whole DNA into small pieces. That is called as shrinking. First, we want to take the DNA from the microbe or any species, and we want to cut down. Then adapter ligation. Why we want to do the adapter ligation? Ligation means that is attachment. We want to add the adapters at the cutted end. Okay, during the restriction digestion, there are the blend ends are there, not a sticky ends are there. Okay, that are not even edges. So we want to add adapters for the ligation. Then library amplification. Then library amplification means this if a small quantity of number of DNA not able to recognize with the or the probe molecule, so we want to amplify means it's increasing the count of the particular DNA. Then sequencing machines are there, and then that sequencing machines are helpful to this find out the proper sequence, no not annotations. Then assembly. After getting the sequence assembly, we want to assemble in order. Assembling means there are small pieces of DNAs in the sample. That's so that adapters, like, okay, they are added the adapters in the third step, right? That adapters are matching the overlapping. This overlapping process, it's the assembling process. Then finishing, Annotation means it's a labeling, it's a marking, then submission to GenBank. These are the step by step process. The next generation sequencing is very important thing, step. First step is DNA extraction, then shearing. And shearing means shearing. In the shearing, they are cut down the DNA into small pieces. That is the restriction digestion. Then adapter ligation. There are not uneven ends are there in digested DNA. So we want to get a proper equal length, that is proper DNA, so they are adding the adapter ligation, library amplification using PCR, then sequencing and assembling, finishing, and then annotation, and then submitted to GenBank. These are the sequential process. Okay, in this next generation sequencing, there are nowadays there are many techniques in the next generation. In that, three techniques are most widely used in the research. That things are Illumina sequencing. This Illumina sequencing, it's helpful for the DNA basis identification, that having unique fluorescent signals. Using the fluorescent signals, they are find out the sequence, that is Illumina. Roche 454 sequencing. Roche ROCHE 454 sequencing. This sequencing technique is called as the pyrosequencing. Pyrosequencing, it's a technique which detect the pyrophosphorylated release and the during the fluorescence lighting. Okay, they are finding their pyrophosphate release. Using that, they are find out the sequences. This nucleosides are incorporated with the polymerase enzyme, right? That every nucleotides are after the polymerase enzyme activity only, they are having a new strand synthesis. So this strand synthesis, it's based on the new strand synthesis, they are find out the opposite nucleotide, that is the sequence identification. Then ion torrent proton sequencing. Ion torrent proton sequencing, it's a rare technique. It's not widely used in the laboratory scale. In this ion torrent sequencing measure the release of H plus ions from the incorporation of that nucleotides in the polymerase. While the polymerase attachment on the the opposite strand with the help of polymerase enzyme, this reaction releases the H plus ions. The release of this H plus ions, they are monitored by the instrument that is called as ion torrent protons sequencing. These are all the about the next generation sequencing. Next one is next question. Investigator discover a new receptor for an unknown ligand and wanted to identify the binding partner of the receptor. They are finding out the ligand. They know the receptor, new receptor. They discover the new receptor. They want to know the binding partner, that is substrate or cofactors, co-receptors. Anti-receptors antibody is not available, but anti-GFP antibody is available. Okay, this which one of the following strategy is most likely identifying the core receptor? One thing we want to know, in this past 3 days, including today, we are discussing a lot of the methods. Okay, this methods in biology is very important, so study the, here also they are asking the method or strategy to identify the core receptors. Here right answer, here their right answer is the receptor is clone as a fusion protein of GFP and expressed in a stimulated cells. The immune precipitated complex obtained by anti-GFP antibody was analyzed by the LCMS and by MS. Okay, this is the concept. Using the final concept, they are first expressed with GFP, then they are analyzed by the LCMS. Next is the set of neonodal mice. Neonodal mice is divided into 4 groups. They are grouped where there are neonodal mice are there. Then they are grouped into 4 groups. One group as neonated were not primed with any antigen. Here, one thing we want to know: this neonated mice, this neonated mice. They are in the research laboratories. They are using this mice for their research. Why this is very important? Why this kind of mice are used means they are this. Day of on particular days, okay, this various they neonodal mice are having similar age related studies between human and the mice. Okay, this neonodal mice are resembles the human ages. And one more thing about this neonodal mice is helpful for their appearance and behaviors. They are expressing the functions of each and every genes. Next thing in the question here, they are primed first. That is so that is able to easily helpful for the functional studies. In this group one, the neonates were not primed with any antigen. So there is no antigen input. The group two neonates were primed with a KLH. Group 3 is neonated with a primed with KLH, but they are additionally added with thymactomice. Thymactomice, we all know the thymus removed mice. Group 4 neonated were KLH primed and thymactomized, but reconstituted with KLH specific CD4 T cells. Thymus is responsible for the production of the CD4 cells, but in this first group, third group, they are thymectomized, but there is no production of T cells. But in the fourth group, they are remove the thymus and additionally they are added this T cells, okay, specific T cells, so it will get immunity. All these mice, when grown adult, when challenged with KLH and anti-KLH IgG antibody was measured in sera. Which of the following is a correct order of magnitude of antibody response? Which okay, how finally they are asking in which mice the antibody production has become high. So here the D is answer. Group 4 is a higher one because group 4 having the additional T cells. So there is a higher number of production in the antibody. So they are added the immune cells. So that in the sorry here okay, they're reconstituted with the CD4 cells. So antibody production become high. Then in a normal cell there is no antigen, so normal antibody is there. So this is a normal antibody quantity. Then neonated mice 2 that is primed with KLH. Okay, there is no not effective antibody production. So there is no antibody production. And here also KLH is thymectomized. Thymus removed mice don't have any function, any immune functions. So that is the really none of the expression in the antibody level. So only they are mentioned. It's more or less similar. Okay, fine. Next question. In order to clone a eukaryotic gene in a PB322 plasmid vector, this is very important while you are studying the genetic engineering. First, we want to know the bacterial lambda phage and PBR322. These three concepts are very important. In the eukaryotic gene PBR322 plasmid vector, the desired DNA fragment was produced by PST1 cleavage is the site. Okay, cleavage site is there in a plasmid. It's incubated with PST digested PBR322 and ligated. Mixed here they mention the PST cleavage site is live within the amplicin resistance gene. Amplicin resistant gene. While you are cutting this, while you are ligated anything with this site, PST1 site, their amplification resistant gene become inactivated. They are not able to express. It's a keyword here. The mixture of ligated cells were used to transform E. coli. The plasmid containing bacteria were selected by their growth in a tetracycline containing medium. Here the main thing, they are tetracycline containing medium. So the tetracycline containing medium is antibiotic. Tetracycline is antibiotic. So this tetracycline resistant gene, it's in active mode. They won't disturb that tetracycline resistance gene. They only disturb the ampicillin resistance gene. Which type of plasmids will be well found? Here the answer is C. Circular PBR322 plasmid containing target genes and resistance genes to apply the to only tetracycline. Circulated PBR322 resistance to both ampicillin and tetracycline, and concatamized PBR322 resistance for both ampicillin and tetracycline. Okay, they are having the resistance gene resistance for the ampicillin and tetracycline, because as per their statement they are having the one cleavage site at the PST1, for the PST1 at the site of ampicillin resistance gene, but they are commonly having the common resistance to resistance to the this ampicillin and the tetracycline. Next question is a fluorophore when transferred from solvent A to B results in the increases in number of vibrational state in a ground state without any changes in the mean energy of either the ground or excited state. What would be the changes seen in the fluorophore emission spectrum? Here the expression level is an increases in the emission bandwidth. This is all about the fluorophore excitation emission related study. Here the as per their statement they are mentioned that fluorophore is transferred from solvent A to B in an increase in the number of vibrational state in the ground state without any changes in the mean energy of either a ground or excited state. So what is would be the change being seen in the fluorophore emission spectrum? The stage is increase in emission bandwidth. Next question is the you wish you wish to localize a given gene product to subcellular level showing the immunofluorescent staining. A routine microscope could not resolve whether the gene product is localized inside the nuclear or on the nuclear membrane. Which of the following will resolve the um, an, unambiguously? Okay, they are mentioned unambiguously means they are not specific. It's the elaborated one. In this case, their answer, which is the best resolve, given the best resolve is the Q and R. A simulation of 3D picture following confocal microscopy and optical sectioning and observing each section. Okay, this is observation each session. This is the concept behind the in the fluorescence immunofluorescence stain. Okay, this concept, yesterday we discussed the that facts concept as once again the basic thing. During apoptosis, the phosphatidyl serine PS is usually present in the inner leaflet of plasma membrane, flip to outer membrane. That is a flipping process. So, during the apoptosis, that PS is flipped to usually present in inlet. In the apoptosis process, that is flipped to outer membrane. This annexin 5 protein is binds to PS. This annexin 5 binds to this PS using the tool as a tool. We identifying the apoptosis cells from necrotic and normal cell population by FACS analysis, fluorescence automated cell counters using the flow cytometric analysis using FITC tagged annexin. FITC means they are mentioned fluorescence tag that is tagged with annexin V. Propydium iodide (PI) is used to stain the nucleus, which usually identified necrotic and late apoptotic cells. So, two things we want to know. If the apoptosis happen means that annexin labeled PS become it's comes to flips to outer membrane. Another case, if that is a late apoptosis, means the PI, the propydium iodide. Here they mention propydium iodide is used to stain the nucleus, which generally identified necrotic and late apoptotic cells. Which of the following area of plot you should get earlier apoptosis cells by FACS analysis? Here, the mention they want to understand the earlier apoptosis. So here that FITC is about the annexion. Then annexion means that we want to know which is the process which they asked the earlier apoptosis. Earlier apoptosis means that is the flips to outer membrane. That flips to outer membrane means that is annexion labeled PS. The annexion is labeled with FITC. So here the case is quadrant 3. So answer is quadrant 3. The next question, muscle tone was increased after electrolytic lesion of quartet nucleus in the cat. The muscle tone decreased with the 7 days. Following explanation were given by researchers. What is the reason means? That is Q&R. A brain tissue surrounding a lesioned area was non-functional due to the circulatory insufficiency immediately after surgery, which leads to greater functional loss. Another option is circulatory status in the surrounding tissues recovered with time, resulting in a partial functional recovery. So this two was reason for the that increase after that muscle tone increase after the electrolytic lesions. Very easiest question. Biologists randomly sampled 3,000 insects from newly found Iceland. It's a new Iceland. In distribution of one thing I want to let you know. This Iceland based question, without this Iceland based question, you would never seen any CSIR question. At least nearly 2 or 3 Iceland population based questions are there. That is inherent biology subject. That was very easiest question also. Just logical questions. The distribution of the abundance sample was in the figure given. Basis count a number of individuals in the sample. Okay, so this is one sample, the individual samples are there. The following is correctly inferred the graph. Which is a correctly inferred graph? D1 is the inferred graph. All species from the Icelands may not be represented by in a sample. In the all samples not able to be represent in a sample. Because why? Because here they mention 2 to 3, 4 to 5. That is not an individual. Okay, they're not individual. This one alone become individual. Because humans. Okay, for example, in one particular village, Chennai having a humans this number for 10 number of humans. But in the animals, okay, dogs, animals groups, that group dogs, cows, and other animals are in a particular count. This is not an individual population. So only they are all species from the Icelands may not be represented in a sample. They are not represented in a sample. Okay, this is all about the methods in biology. Part C questions. Tomorrow onwards we will start the unit 1 questions. Thank you everyone. I welcome you all to tomorrow class, day 21 class. Please try to join in all classes. Thank you. If you have any queries, you can ask in